Optimal media for human pluripotent stem cells. Pluripotent stem cells are changing the face of regenerative medicine and drug discovery, and there's no better evidence than their ever-growing role in biomedical research. This stresses the vital need for an optimal medium that would maximize their growth and safety. Our library of mutated haploid human embryonic stem cells and our advanced analysis of its composition over time allows us to detect mutations that either become eliminated, indicating the mutation occurred in a gene that's essential for cell growth, or take over the culture, indicating the mutation occurred in a gene that restricts cell growth. These findings highlight pathways crucial for human pluripotent stem cells and enable the identification of novel medium components that would maximize the growth of pluripotent cells in culture while maintaining their genomic integrity, addressing one of today's most crucial requirements of the life sciences industry and academia, and brings new means for drug development. To model specific diseases, we genetically modify haploid human embryonic stem cells and induce the appearance of a disease phenotype. We then use these cells to generate a mutant library of cells that share a common disease hallmark. These disease model-based libraries can be screened for cells in which the disease phenotype is corrected, enabling the identification of genetic interactions that can serve as new drug targets, enabling novel therapies for human disorders. This strategy provides a revolutionary framework to help fight cancer, enable the reversion of a genetic disorder and treatment of other unmet clinical needs, all by identifying the critical genes and pathways involved. New approach for reproductive medicine. Having their own child is a desire shared by many couples. However, infertility, which occurs in more than 10% of couples, as well as gender issues, prevents many from fulfilling their dream. Human embryonic stem cells have the potential to differentiate into all cell types in the body, including reproductive egg cells. To differentiate into a fully mature egg, a progenitor cell must first reduce its number of chromosomes by half through a process called meiosis and become a haploid cell. Although meiosis occurs naturally in our body, it is still challenging to mimic in the lab. Using haploid human embryonic stem cells from female and male origin, which already comprise half of the genome, will bypass the need for meiosis. This could potentially facilitate the in vitro generation of human eggs carrying female or male genetics and revolutionize reproductive medicine. Establishing egg reservoirs would allow innovative research and medical opportunities for treating infertility and fertility preservation as never before.